Hello students, this is Praveen Trivedi and we have been discussing regarding Unit 5, Fundamental Unit of Life. So, we have started discussing right from the beginning that regarding and plasma membrane, we reach up to the plasma membrane. And so here, what are the different mechanisms? Actually, plasma membrane is involving in the exchange of material in and out of the cell. And what actually the principles which have been involved in the movement of material in and out of the cell, let us see regarding that. And we have started discussing them. We have already discussed regarding diffusion, we have discussed regarding osmosis, and then active transport also yesterday we have discussed. So diffusion is the movement of material from region of higher concentration to the lower concentration. Similarly, the process was happening in the osmosis also. And then we have discussed active transport. Diffusion and osmosis, they both are the passive processes, whereas there is no any use of the energy, only the kinetic energy of the material or the molecules moving in and out is sufficient for getting the regulation of movement, for facilitating the movement. Whereas in the case of active transport, we have seen that there is a utilization of energy, that is in the form of ATP, which has been used because the material have to move from an area of lower concentration to the area of higher concentration against the concentration gradient. And now students, this is the last, we can say, the mechanism which is involving in the movement of material through plasma membrane, and that is said to be endocytosis. So endocytosis, it is actually the process of taking in and releasing out, or we can say the exchange of material in and out of the cell, that is said to be endocytosis. So material, particularly the food particles, here particularly the food particles are moving in of the cell, and then the digested remnants of that food particle will be released out or thrown out. So endocytosis, process of moving in, getting the material moving in and releasing out, is known as endocytosis. The material may be some of the time food, or maybe some of the time some other organisms also. We shall be discussing in detail. So through plasma membrane. Now what is the specific name given to the material moving in and out? That is called as macrobiomolecule. The macro word has been added here as they are larger in the size as compared to the material and molecules, particles moving by the other processes like osmosis, diffusion, etc. Okay, now plasma membrane. Now how this particular endocytosis has been, we can say, facilitated. Why? Because the plasma membrane is having the specific capacity of flexible. We can say it is flexible in nature, having the capacity of flexibility. And due to this flexibility, what is happening? The plasma membrane get fold and unfold. This is actually the mechanism. The physical, we can say, the mechanism takes place is actually the folding and unfolding of the plasma membrane. And due to that folding and unfolding of the plasma membrane, the material will get enter into the cell. So we shall be discussing this process in detail in the next slide. So taking of the material in is called as endocytosis, whereas the throwing out of the digested, we can say, the hydrolyzed material, hydrolyzed or digested remnants, is actually known as exocytosis. So endo means entry and exo means when it is moving out. So plasma, this particular endocytosis, we can say, can be divided into two types, exocytosis and endocytosis. This we have already discussed. Then what are actually the principle involving in the governing of movement? That is folding and unfolding of plasma membrane. And folding and unfolding has been facilitated. How the folding and unfolding has been facilitated? Folding and unfolding has been facilitated due to the flexibility in plasma membrane. So this is the flexibility only, which is just we can say governing the movement of material in and out of the cell. So now movement of solid particles. Now again, as endocytosis has been divided into two types, exocytosis and endocytosis. Endo means entry, exo means moving out. Similarly, when the particles are solid in nature, this is to be discussed again, when the particles are solid in nature, then the process is known as phagocytosis. This is phagocytosis. And whenever the material which have been entering into the cell by the process of endocytosis, then the process is known as pinocytosis. So movement of solid particles facilitated by phagocytosis, movement of liquid particles being facilitated by pinocytosis. This is also one of the division of process of endocytosis. So endocytosis exocytosis and endocytosis. And then movement of, if we can say, suppose the particles are solid, then it is known as phago, and when the we can say the material or particles are in the liquid, they will get dissolved in the extracellular fluid. If suppose the materials are there, so some of the very smaller materials are getting dissolved into the extracellular fluid and then getting entered into the cell by the process of pinocytosis. Now here phago, the cells which are facilitating such processes, the cells which are facilitating, which are governing the process of, we can say, endocytosis, are called as the cells are known as phagocytes, where the phago means to eat. What does this phago word means? Phago means eating. So such cells are known as phagocytes. Now the cellular component inside the cell, which is known as phagosome or vacuole, we shall be discussing regarding that vacuole or phagosome when we start going to discuss from tomorrow regarding the cell organelle. Then the vacuole will be there. So what that vacuole is? Vacuole is also called as phagosome, and it is there inside the cell. As soon as the material will get entered through the plasma membrane in the cell, the material will be getting embedded inside the vacuole, and this vacuole is containing the digestive or destructive enzyme which leads the digestion or destruction of the material which has been entered. And then what happens due to that destructive enzyme, solid versus vesicle, and whereas here also there is a division. If the particular material is solid, then the vacuole will be participating in the digestion or destruction of the material. And if that particular material which has been entered is liquid in nature, if suppose it is liquid in nature, then instead of vacuole there are certain other organelle present, that is called as vesicles. These vesicles will be participating in the digestion of that liquid particle or liquid materials. So number one, endocytosis, exocytosis, phagocytosis, pinocytosis, and then the new phagosomes. Okay, phagosomes, that is vacuole or vesicles. So this is regarding the overall movement of the material by the process of endocytosis. This is also one of the principles being governed by plasma membrane, and this is also facilitating the movement of particle from outside of the cell to the inside of the cell. Now we can just see a diagrammatic representation of the process of that we can say the endocytosis by the we can say amoeba. Now see, endocytosis in amoeba, how does it takes place? Now if we will just observe the diagram of that amoeba, now this is the what happened, this is amoeba, and this is the folding of plasma membrane. Now you can see here there is a sac like structure have been formed. Whenever the black color is material, that is a we can say that is one of the particulate material, that means solid material. Now see here, that is a particulate material, so that is a solid material. When that solid material comes in the contact of the edges of that plasma membrane, due to flexibility, plasma membrane starts folding, and due to the folding there will be a formation of, we can say, the sac like structure. Now the particle will get entered into the sac. When the particle once will get entered into the sac, then what happen? The upper edges of that plasma membrane again reaches towards each other, and what happen? The plasma membrane again attain the regular structure, the routine structure of the plasma membrane, the initial structure, and material will get entered. And now here you can see the material has been embedded in a circular structure. That structure is called as vacuole. And here inside this particular structure there will be the release. This is a single membranous structure. Vacuole is single membranous structure. So what happens? When the destructive enzymes will be released inside that vacuole, that particular material, now see here the name phagosome has been given. Phagosome, it is also called as vacuole. So inside that vacuole there will be the release of certain enzymes, and due to the action of that enzymes on the particulate material, the material will be broken down, and we can say the digested or destroyed, and then the remnants will again move out in the same way. So what happens? This vacuole will get again. So see here one more example I have collected for you for the same process of endocytosis. You can just go through that. Now see, here is the one of the another example of, we can say, the process of endocytosis. So what is happening here? Here again the outside particles, the foreign particles are there. As soon as these foreign particles comes in the contact of the wall of plasma membrane, the plasma membrane has made one fold, and inside that fold the material will get entered. As soon as the material will get entered into the fold will get closes, and the material get inside the cell. When the material will get inside the cell, it has been surrounded by, or we can say it get entered into the vacuole. Then again there will be the release of the enzyme. Here you can see the breakdown of that material. Now see, this is the complete material, this is again the complete material. Then this material has been acted upon by the release of enzymes here inside the vacuole, the destructive or digestive enzyme. Then the material has been broken down into small pieces. Again the more smaller pieces have been, we can say, released, and immediately after the complete digestion, after that complete cycle, what happened, the material will reach again to the towards the plasma membrane. This vacuole will be getting open, and this particular material has been released again by the, we can say, the opening of plasma membrane. So this is the complete cycle will takes place. Material will come in the contact of plasma membrane, get entered and get acted upon by the destructive enzymes, and then again broken down into very smaller pieces. The material required will be released into the cell for utilization, and then the material, the digested, the remnants will be moving out. So this is actually the process of endocytosis and exocytosis. This is actually the process which is involving in, moreover, you can see the WBC, whatever be the WBC molecules are there inside the body, and we say that WBC are actually the soldiers of our body. How we are defining the WBC? We are defining the WBC as the soldiers of the body. So what they are doing, as soon as any microbe will enter into the body or into the blood of our body, any virus, bacteria or any of the disease causing microorganism, then what happen, inside our body also the same process happens, because the WBC will get acted upon. WBC will get acted upon, because what happen, as soon as that particular virus or bacteria will come in the contact of the plasma membrane of a WBC, because our WBC is also a cell that is said to be white blood cells. So as soon as that particular disease organism will come in the contact of the plasma membrane of our WBC, immediately the folding of the WBC will take place, plasma membrane will take place, the microbe will get entered into that WBC cell, and the same process will be happening as we are discussing just now. The same process will happening in the same way and causing the destruction of that virus, and that is why they have been told to be the soldiers of our body. Similarly, suppose this is the microbe, suppose this is the WBC, microbe will come in the contact of the wall of our WBC. Then what happens, folding takes place, microbe will get enter into the vacuole will be releasing the destructive enzyme will digest that microbe, and the remnants will again get out of the body. And at the same time, when such a kind of the, we can say, the war will be taking place inside our blood cells, that is WBC are available in the blood. So when such a kind of the war will be taking place, you people might have felt the fever, the temperature of the body increases. Why? Because actually this is happening, and this will increase the temperature of our body. So these are some of the mechanisms we shall discuss in higher classes in detail. So this is actually the overall mechanism taken place governed by plasma membrane